Welcome everyone to the second lecture of the third module on ocean ecosystems and climate. As usual, please post your questions in the chat during the lecture. We will collate them and then ask the questions after the lecture. And as TJ mentioned, everything is recorded and you can find the lectures on the YouTube channel. So it's a pleasure to introduce Dr. Bob Bruin for today's lecture. He's a senior lecturer in physical geography at the University of Exeter in the UK. After completing his PhD in marine remote sensing at the University of Plymouth, Bob has worked for 8 years as a marine scientist in the remote sensing group at Plymouth Marine Laboratory and for the UK National Centre for Earth Observation. He joined the University of Exeter in 2019 as a lecturer and is now a senior lecturer and also a UKRI Future Leader Fellow. Bob's research is focused on the use of satellite data for understanding marine biogeochemistry, and his interests include marine optics, marine primary production, marine ecology, bio-optical modeling, and developing algorithms for remote sensing of phytoplankton groups. He's also interested in using citizen science to expand observations of key environmental indicators, such as water temperature and water quality in undersampled regions. And he has joined some of the discussions on that in the previous lectures. Bob's work spans global and regional scales, ranging from lakes to open oceans and from tropical to polar waters. And today he will be talking about primary production. The floor is yours, Bob.
Thank you, Gemma. That was a very nice introduction. Okay, I'm just going to share my screen. And do let me know if you can see my slides.
Yes, perfect.
All good?
Yeah.
Okay, well, welcome to this lecture. It's a real pleasure to talk about this subject. On the one hand, I'm a little bit nervous because there are various people in the panel, Shuba, Gemma and Tom in particular, but also Osvaldo and Lauren knows a bit as well, who are probably even better qualified to give this lecture than I am. But certainly primary production is a topic that I've become increasingly interested in over the years. And it's a real pleasure to talk about this during this seminar series. And in particular, because Trevor Platt was a pioneer in quantifying and understanding primary production by phytoplankton in the sea at various scales in space and time. And so I just hope I can do some justice to the topic, given that this seminar series is part of the TPS Foundation. Okay, so the outline of this lecture, I'm going to keep it fairly simple because I'm a fairly simple guy, I guess. But I'm going to start by really talking first about phytoplankton biomass. Excuse me, I'm just going to shut my window because I've got a little bit of noise coming through from outside. There we go. Okay, so I'm going to talk about phytoplankton biomass and define what that is. How we measure that at sea and how we measure it from space, which will link back to a number of other modules in this training course. Before I then move on to primary production, and the reasons for this I'll go into when we talk about primary production, but it's often useful to look at primary production as a biomass normalized variable, particularly when we're modeling it from satellite observations. I'm then going to talk about the importance of primary production. So why is it so important and why are we doing this? Briefly touching on how we make measurements of primary production in the sea, and then from satellite observations, and then finishing by highlighting a number of resources that are available to the community, data sets that are available that you are free to play with afterwards. Okay, so like all my lectures at university, I like to sort of define some learning outcomes. So I hope that after this lecture, you have a good grasp of what primary production is, firstly, understanding the differences between phytoplankton biomass and primary production, understand how we measure it at sea and from space, and understand where you can get some satellite data from primary production should you be interested in pursuing this topic in the future. Okay, so starting with the definition of phytoplankton biomass. So the biomass of phytoplankton can be described as its sort of local abundance. The equivalent term in fisheries is often used as stock size. So it's sort of a snapshot in time of the amount of phytoplankton in the water. Now, many metrics exist for phytoplankton biomass. Two commonly used metrics are carbon, but also nitrogen as well, because a lot of modelers like to work in flows of nitrogen in their ecosystem models. Carbon is a useful metric because it's an abundant element. If we look at the Redfield ratio, for instance, there's a lot more carbon than there is nitrogen or phosphate. However, there are some disadvantages with the metric of carbon in that it can be quite tricky to measure directly in the ocean. Now, in terms of phytoplankton biomass, it's interesting often to compare the stock of biomass or the amount of biomass of phytoplankton in the ocean with the amount of biomass of terrestrial plants on land. And if we do that, we can see that actually the biomass of phytoplankton in a snapshot of time in the ocean is minuscule almost compared to the amount of biomass of plants on land, terrestrial plants here. But that, as I'll allude to a little bit later on, doesn't in any way mean that phytoplankton are not important for primary production. Now, in terms of a common index used for phytoplankton biomass, and it's an index that I'll continue to use throughout this lecture, is that of the chlorophyll A concentration. The concentration of chlorophyll A pigments, so photosynthetic pigments in phytoplankton, is often used as an index of phytoplankton biomass. The reason being, well, a number of reasons. Firstly, it can be associated directly to phytoplankton in the ocean, unlike carbon, which is in bacteria, it's in zooplankton. And so it's a fairly easy metric to measure in the ocean, and also it has a direct link to ocean colour and consequently how we can observe phytoplankton biomass from space. Okay, so how do we measure chlorophyll A concentration in the sea? Well, there are a number of ways of doing this. Often it requires going out into the ocean and collecting water. Now that can either be from a CTD rosette, so you lower the CTD vertically throughout the water and you snap the Niskin bottles shut at various depths, and you can take that water on board and you can filter it, and then you can put the filters into some chemicals to extract the chlorophyll concentration, and then you can measure that in a pherometer. That's one way of doing it. Another way of measuring chlorophyll using fluorometry is to do it in vivo. So to measure it directly, either using a fluorometer attached to the CTD or potentially on a underway, non-toxic underway sample. So if you look at this image on the top left here, this is just sort of illustrating how water can be pumped through a ship as it moves, and you can continuously measure chlorophyll fluorescence. Now, there are advantages and disadvantages to both these techniques. Typically, the in vitro method, the extraction method, is a lot more accurate for measuring chlorophyll in the ocean and phytoplankton biomass. Another way of doing it, which is commonly adopted in satellite validation protocols, is to use high-performance liquid chromatography. So similar to the fluorometric methods, you filter onto a GFF or glass fiber filter. you typically flash freeze it in liquid nitrogen, put it in the -80 freezer, and then when you have the time, you can take that sample out, you can put it in some chemicals to extract the pigments, and then use column chromatography in order to separate out those pigments and measure them using high performance liquid chromatography. This way you can also look at other pigments in the phytoplankton sample, which is useful for looking at the phytoplankton composition or looking at the physiological state of the phytoplankton in the water. But this is a common technique that's used to measure chlorophyll concentration, the metric of phytoplankton biomass that we're going to be using in this lecture. Now, more recently, or in more recent years, another technique is increasingly being adopted, and that is in vivo spectrophotometry. So here you use a spectrophotometer sort of inline. So this is an example, I think, on the bottom left of an ACS, where water is can be pumped through the ACS, and it measures the absorption coefficient of particles if you do filter no filter system, and you can get very accurate measurements of particulate absorption as a function of wavelength, shown on the top right there. And of course you have this absorption bump in the red, which is directly related to the chlorophyll A pigment. And if you look at the height of this bump, it's very tightly correlated with the chlorophyll concentration. So more recently, people have been attaching these types of instruments onto the CTD rosette to measure profiles of chlorophyll in the ocean, but also attaching them to the non-toxic underway water supply to get very accurate continuous measurements of chlorophyll A phytoplankton biomass as the ship moves through the ocean. Okay, so that's a little bit about how typically we measure phytoplankton chlorophyll A concentration at sea. Now, in terms of how we measure the chlorophyll concentration from space, I know some of this will touch on previous training modules like Module 2 here, which gave us an introduction to ocean colour. But how we do it from satellite is simply by looking at the reflected light from the ocean surface and then relating that to the chlorophyll A pigment. Now, chlorophyll A absorbs very strongly in the blue and a little bit in the red. So what generally happens is the more chlorophyll concentration in the water, the more blue light gets absorbed, and hence that impacts the reflected light that the satellite sees, and the oceans begin to look greener. So this is an example of an image actually took from my phone in the center of the North Atlantic gyre. And as you can see in this image, the waters look very blue, and this is because there is not very much phytoplankton biomass in the water. Chlorophyll concentrations are very low, so the optical properties of pure water dominate, and the reflected light seen from a phone or seen from a satellite looks very blue. As the chlorophyll concentrations increase, this is another photo taken in very high chlorophyll waters. We can see that a lot of that blue light gets absorbed, and the oceans begin to look green. And so by looking at spectral variations in the remote sensing reflectance measured from the satellite, we can get pretty good approximations of the chlorophyll concentration. And that's just shown again in an example here. So this is from one of Schuber's papers published back in 2017. It's a very large in situ database of the chlorophyll concentration shown on the y-axis here, plotted as a function of essentially the ratio of blue to green lights. This is the maximum band ratio. So basically what's plotted on the x-axis here is a ratio of blue to green light, but it's switching between the blue wavelengths 443 to 490 to 510, depending on which wavelength has the highest signal. And so what you can see in this image is that the chlorophyll concentration is quite well correlated with the ratio of blue to green light. Now, there's a lot of scatter around these relationships, which are down to a number of factors, some of which you possibly touched on, maybe when you were looking at the module or listening to the module on optically complex waters, where we know that this ratio of blue to green light is also sensitive to other constituents in the water. So if there's a lot of CDOM in the water, for the same chlorophyll concentration, it's going to modify that ratio. If there's low amounts of CDOM, it's going to affect it. If there's high amounts of non-algal particles, and so on and so forth. So it's a model that's very much based on what they used to call a case one assumption, where phytoplankton are the principal component in the ocean controlling the optical properties. So some of the first satellite observations of chlorophyll A concentration came with the launch of the Coastal Zone Colour Scanner by NASA in the 1970s. And sort of illustrated in these two publications by Hovis and Gordon in Science in 1980. And this was really revolutionary at the time. First examples of how we can, or among the first examples of how we can use satellite observations to monitor the biomass of phytoplankton over large scales. So you can see here where Florida is just in the image here. And on the right-hand side, the image was matched alongside a transect of chlorophyll measurements collected by a ship using fluorometric-based measurements. And you can see how well correlated the spatial changes in chlorophyll are when matched between the satellite and the ship. Okay, in more recent years, we've now are blessed with a suite of very highly accurate and well-calibrated satellite ocean color missions. These are just some examples of Sentinel-3 in the top left, MODIS Aqua and VIRS. And as you can see in this nice image in the middle, how the spectral variations or the color observed by these satellites can be linked to the concentration or the biomass of phytoplankton in the water in the context of the chlorophyll concentration. Those very green waters being seeing the presence of much higher phytoplankton abundance, a lot more chlorophyll in the water than the blue regions. And I pinched this slide from Tom Jackson's talk last week, but now we're blessed with nearly 25 years of continuous ocean color observations, and with projects like the Ocean Color Climate Change Initiative, where they've strived to successfully merge many different satellite ocean color missions together to produce this continuous time series of chlorophyll concentrations. We can really get detailed patterns of how the phytoplankton biomass in the ocean changes seasonally, changes spatially, and also changes at interannual timescales. And that's sort of illustrated in this figure here. So this is a figure taken from a paper published back in 2016. And we're just focusing on two different regions of the ocean here, the equatorial Pacific on the top part of the image, and also the Indian Ocean on the bottom. And what we've plotted here are climate indices. On the top right is the multivariate El Nino La Nina Southern Oscillation Index as a function of time over the periods of 1980 to 2020. And on the bottom is the Indian Ocean dipole, which is essentially a difference in sea surface temperature between the two boxes shown in the bottom left. And what we've done is we've overlaid the time series of chlorophyll concentrations. This is again from the Ocean Colour Climate Change Initiative. This was an earlier version of the data set. And what you can see is that interannual variations in climate are very tightly coupled to the chlorophyll concentration, the biomass of phytoplankton in the water. And one of the reasons this is because the lifespans of phytoplankton are relatively short compared to plants on land, and so they are capable of responding very rapidly to variations in climate. Okay, so that was a bit about phytoplankton biomass. So getting kind of into the meat now of the lecture, I'm going to begin talking a bit about primary production now. What is primary production? Well, primary production is basically the rate of photosynthesis of phytoplankton. It's the equivalent term in the terms of fisheries is sort of the reproduction and growth of the fish. I like this term. I think that was coined by Schubert and Trevor a while back, saying that if biomass is like money in the bank, primary production is like the interest earned by the capital. So it's a sort of nice way of separating out what biomass and primary production is. Now, one of the very interesting things with phytoplankton, if you recall earlier on in this lecture series, we talked about how the biomass of phytoplankton is minuscule compared to the biomass of all plants on land. Well, when it comes to primary production, things are very different. Primary production by phytoplankton is pretty much equivalent in the ocean to that by terrestrial plants on land. So both of which contribute around 50 gigatons of carbon to global primary production. Now, the reason this is, again, comes back to a similar point I made in the in an earlier slide, that phytoplankton have very short growth cycles. They're constantly growing and dying, whereas the life cycles of terrestrial plants are much longer. And as a result, the primary production of phytoplankton is equivalent, or thought to be equivalent, to that of terrestrial plants on land. So they are a major player in a global planetary primary production, as illustrated by some of these references shown on the bottom there. So what is primary production? Well, we talked about the rate of photosynthesis. So what is that? Well, it's basically the conversion of inorganic carbon from carbon dioxide into organic carbon with light as the energy source. So in the ocean, this is typically expressed in terms of mass of carbon converted or fixed per unit volume of water per unit time. So it's a rate. And if we look at the basic photosynthesis equation and how it works, we can see that we have water, H2O, and we have CO2. We use the available energy in the form of sunlight that's absorbed by the phytoplankton through the process of photosynthesis. And these elements and compounds are converted into carbohydrates, stored energy, plus oxygen. So oxygen is produced and dissipated energy. So that's the sort of basic equation of photosynthesis. So like plants on land, the growth requirements of phytoplankton are similar. They require water, which of course is extremely abundant in the ocean, not a problem. They also require carbon dioxide. Again, the carbon dioxide concentrations in the ocean are quite high, and so that's available. They need light. Now that potentially becomes a bit of a problem because light gets attenuated quite quickly in the ocean. So phytoplankton are only able to grow where there is available light at the surface of the ocean. And like the plants on land, they require nutrients, not only macronutrients like nitrogen and phosphorus, but they also require micronutrients. So essential nutrients like iron, even though they only require low concentrations of iron, they're very important in the process of photosynthesis. Okay, so a few definitions before we move on of primary production, because there are various aspects of primary production in the ocean. The first one I'm going to talk about is gross primary production. So this is often defined as the total rate of organic carbon produced by autotrophs or by phytoplankton. So it's the total amount of primary production. Then there's net primary production. So this is referred to as the total rate of organic carbon production by phytoplankton, by autotrophs, but minus the respiration by phytoplankton. So respiration is, of course, the inverse process of photosynthesis, whereby oxygen is converted back into CO2. So with net primary production, you subtract the respiration away from the gross primary production in order to get net primary production, but the respiration only by phytoplankton. Now, another term commonly used is that of net community production, and that is defined as the total rate of organic carbon production by autotrophs by phytoplankton minus the respiration by not only phytoplankton, but all the organisms in the ecosystem. And another term we often refer to is export production, which is defined as the amount of organic matter produced by primary production in the surface ocean that is lost to the deep ocean. So I thought it's important just to make some definitions of those terms before we move on. I think I saw a question pop up about that as well, so it was quite good timing. Okay, so what is this process of primary production then, and why is it important? Well, this is a sort of schematic diagram. I think Tom might have shown this one last week as well, of the ocean carbon cycle, a simplification of the ocean carbon cycle. And where does primary production lie? It's lying right about here in this process. So we have our dissolved inorganic carbon, which is being converted into particular organic carbon through the process of photosynthesis. So this is kind of like our gross primary production. And then you would minus the respiration just by phytoplankton here in order to get your net primary production. Now, this little process in the middle arguably is driving a lot of this much larger ocean carbon cycle. So the production produced by phytoplankton then gets consumed by zooplankton. That can then you then have a whole suite of quite complex processes where bacteria are breaking down some of that carbon. There's a lot of respiration going back in the form of DIC. There are DOCs being produced through bacterial remineralization, and some of this material is then sinking out of the surface layer, forming our export production, either directly by the phytoplankton or aggregating in the form of fecal pellets, for instance, by zooplankton, pooing out a lot of the material that's been eaten. There's also complex vertical migration going on with the zooplankton, where they come up at the surface during the night to feed and then go down to depth and excrete some of that carbon that's been eaten. There's also physical processes moving around these different pools of carbon in the ocean, and there's a whole lot of complex processes going on at depth as well, resulting eventually in an element of that primary production produced in the surface being transferred into the deep ocean in the form of export production. Complex processes going on, but essentially the engine behind the whole of this is the primary production by phytoplankton in the ocean. The part of the image on the right here is just sort of showing various ways we can monitor these pools and fluxes of carbon in the ocean, satellites being a big focus of this talk here. Okay, So why is this primary production important? Well, I sort of just touched on that then, but I'm going to go in a little bit further detail here. There are many reasons why primary production is important, but I'm just going to focus on two key reasons here. Firstly, I'm going to talk about the ocean carbon cycle. So what happens? What happens with phytoplankton in the surface layer? Well, phytoplankton are photosynthesizing. So that's drawing CO2 out of the water, and it's modifying the gradient in CO2 in the ocean. So this is an important point to make first, because this disequilibrium caused by production of phytoplankton at the surface reduces the CO2 concentrations at the surface, which can, of course, promote air-sea gas exchange. Now that primary production produced by phytoplankton at the surface, as we showed in the previous slide, a large portion of that will get respired back from organic carbon into inorganic carbon, so going back to CO2, but a certain amount of it gets exported down into the interior ocean. So it's thought around 50 gigatons of carbon is produced in the surface of the ocean. Around 10% of that gets exported into the interior ocean. And this continues to sink and gets quite heavily remineralized, so converted back into CO2 as it sinks down towards the sea floor. And only about 2 gigatons is thought to be deposited in at the surface, at the bottom of the ocean, and around only 0.2 getting buried into the sediments. But one of the important points to note here is that the further this carbon gets down into the deep ocean, the likelihood is that it will be kept away from re-emission into the atmosphere for a longer period of time. So basically, there's increasing timescales of ocean carbon storage as that particular organic carbon sinks down to the seabed. So this process of primary production and the ocean biological carbon pump is very important in controlling the surface CO2 concentrations and consequently the ability of the ocean to absorb CO2 from the atmosphere. So that's one important reason. Now, another important reason is fisheries, primary production are at the very base of the marine food web, right? So all higher trophic levels are dependent on their productivity. And there's this classical sort of 10% law. They say that 90% of the energy gets dissipated as you go from your primary to your secondary to your tertiary trophic levels. But ultimately, the production by phytoplankton at the very base is critically important for feeding, for sustaining the entire marine ecosystem. And this is all the way through to humans through the seafood we eat. So this is a nice paper I came across, I think last year, and it was some work conducted in the US where they showed how the average annual productivity here, plotted on the x-axis, is well correlated with the fisheries revenue in the US. So the higher the productivity, the larger the fisheries revenue. I mean, of course, there's many other factors that impact fisheries, but it's just to illustrate the importance of primary production in this process. Okay, so on to measuring primary production at sea. Well, one of the first kind of ways people did this, at least from reading a little bit of literature, particularly from this very nice book by Mills in 2012. If you're interested in learning a bit more about the history of biological oceanography, I strongly suggest you read this book here by Eric Mills. It gives a wonderful history of the early biological oceanography. But one of the ways people got an index of phytoplankton productivity originally was to count phytoplankton cells and then make some sort of assumptions about the productivity per cell in order to get an idea of primary production in the ocean. But one of the interesting methods developed, I think, back in the 1930s was by H.H. Gran, a Norwegian scientist, I believe, who made use of the fact that oxygen varies with the abundance of phytoplankton, of course, oxygen being a product of the process of photosynthesis, and so can be used to get quite a quick bulk index of primary production. And so he developed this light and dark bottle method, whereby you would sort of suspend bottles at various depths. Often the bottle right at the bottom you would cover up so you can get some idea of respiration, and then you would monitor each bottle with known concentrations of phytoplankton cells, known salinity and oxygen content, and then you would look at how that would change over a period of time in order to get a proxy of primary production. So this was one of the earlier techniques known as the light and dark bottle method. Now, I wanted to link back to the module 7 by Grints and George on water quality and human health. He talked a bit about Secchi discs in that module as well. So some of the earlier work in the sort of 80s and 90s also, and this was some of the first approaches that looked at how clarity of the water in the ocean can be linked to productivity. Given that there was this long history of Secchi depth observations going back to the late 19th century, there was various work that would relate the Secchi depth to the phytoplankton biomass and then make assumptions about the productivity per unit biomass in order to get an idea of how phytoplankton productivity was changing in the ocean. The paper I just pick out here by Fakowski and Wilson in 1992, which was sort of a response to an earlier paper suggesting that the phytoplankton productivity was increasing in the North Pacific, and they studied the Secchi depths from a long time series and found that actually they weren't seeing any significant changes in Secchi depth, implying that the productivity in the North Pacific wasn't changing drastically. So a link there to some of the earlier work that was taught in this training module. Now, in terms of measuring primary production at sea, in the sort of modern era, there are a whole range of techniques to do it. And I don't really have the time to go into all these different methods. The 14C method, I'll talk about it maybe briefly in a little bit. This is a table that was taken from a recent paper published by Schuber, a book chapter published by led by Schuber in 2019 that lists the various methods we can measure primary production in the ocean. Again, with reference to the different types of primary production: gross, net, and community production, and export production, and on the various timescales that these measurements need to be conducted at in order to get at the production property. In the same book, actually, in the same book, Encyclopedia of Ocean Sciences, there's also another chapter by Church, Cullen and Carl, which also talk about some of these other techniques as well, optical techniques, as well as the sort of standard 14C methods and so on and so forth. So if you're interested in the subject and want to learn a bit more about how we measure this in the sea directly, then I really strongly suggest you have a little look at these two book chapters. The other thing as well is that the IOCCG have just published a protocol series on measuring primary production at sea, volume 7, Aquatic Primary Production Field Protocols. This is really an excellent resource. I strongly recommend you looking at, and they have this nice figure in one of the earlier chapters which talks about each of these different methods of measuring primary production in the ocean and how they vary on temporal and spatial scales. Now, in terms of my experience, I haven't had a lot of experience in measuring primary production directly at sea. I'm normally one of these individuals who likes to play with satellite images instead, but I have been on a few of these Atlantic Meridional Transect cruises, and these are just some of the ways we did it on those cruises on AMT. So this is a figure I actually took from Gavin Tilstone, who would come on board the AMT and do a lot of these on-deck incubations. So what you would do here is you would collect a water sample pre-dawn, before the light, before the sun rises, and then you would incubate them on deck over the course of the day. So you would use the 14C technique, whereby you add 14C to the sample, and basically you run these incubations over the course of about 12 to 24 hours at various light gradients that are designed to reflect the depths at which they were collected in the water. And then you would look at how much of that 14C was taken up in the form of particulate carbon by measuring it on a scintillation counter afterwards, from which you can get approximation of the primary production. Another common technique that's used are much shorter term photosynthesis irradiance experiments run in photosynthotrons, whereby you take your sample and then you incubate them over a light gradient, and you do that normally on a much shorter time period of 1 to 2 hours, and then you, again using the sort of C14 method, you measure the primary production, and then what you typically do is you fit what they call this photosynthesis irradiance curve, which I'm going to start talking about in a minute, whereby the irradiance is plotted on the x-axis as a function of the production on the y-axis. This is production normalized to biomass. So, and then when you plot this curve, you fit what they call a photosynthesis irradiance curve, which again links back to the second slide I showed. Trevor Platt was a real pioneer in how we fit these functions to the data and how we can interpret the parameters of the function. So here we have the assimilation number and the initial slope and IK, which I'll talk about in a moment. Now, one of the slight issues with all these primary production measurements at the sea is often they're measuring slightly different aspects of primary production. The incubations are at different periods of time, so it's very hard to reconcile all of the different techniques for measuring. But I just refer you to a nice paper by Jarko in 2016. So there are some nice work that's being done where they use mathematical modeling to start to more directly relate these different types of primary production measurements. Okay, so that was a bit about measuring primary production in the sea. Now I'm going to talk about how we do it from satellite. So I first wanted to show one of the first maps of global primary production from space. So this was published by Alan Longhurst, Schubert and Trevor and colleagues back in the mid-1990s. And it was really quite a revolutionary study. There were subsequent studies published in 1996 and 1997 by David Antoine and Mike Berenfeld and Fukowski. But these were the first sort of real illustrations of the power of satellite remote sensing, the ability to use satellite ocean color observations to kind of for the first time get very, remarkably accurate estimates of global primary productivity. Using satellite remote sensing directly, but also as an extrapolation tool for our in situ observations. And at the time, they quantified global primary productivity to be around 50 gigatons, 40 to 50 gigatons of carbon per year, which, 25, nearly 30 years later is a number that we're still arriving at with our satellite observations. Okay, so now there are many ways or many different types of models for primary production from space, and I'm going to stick with a very simple one for the last part of this lecture here. But I would just like to, I'll refer you, if you're interested in learning a bit more about these different approaches, to a paper by Schubert and Trevor published in 2007 in Oceanology, which really was a nice piece of work in my opinion, because what it showed is that although there are very different techniques used to measure satellite primary production, essentially they all boil down to the same 3 or 4 variables / parameters, and they are fundamentally similar. So I'm going to use an available light model for the rest of this lecture, which is quite a fairly simple approach, but it incorporates all of the key aspects of modeling primary production from space. And at this point as well, I hope these next few slides illustrate why I separated out phytoplankton biomass from primary production in the earlier part of this lecture. So it's common in computing primary production to work with biomass normalized production, so what they call PB here. And this is just to sort of illustrate that we do that by dividing the primary production by the biomass to give ourselves PB, and we can reconstruct the primary production by knowing the normalized primary production and the biomass. And one of the key reasons we do this is because we can get good, accurate estimates of chlorophyll concentration from space. So we can use our biomass as a scaling factor if we know the normalized primary production, biomass normalized primary production. Okay, so I'm going to now work with a little bit with PB rather than with P. So production normalized by biomass. And as I mentioned in a couple of slides earlier, this is what they call a photosynthesis irradiance curve. Again, a lot of work by Trevor, Platt and colleagues in the 80s and 70s, looking at different ways of quantifying this. But here we have normalized production on the y-axis as a function of irradiance on the x-axis. And if we plot our primary production measurements, we can fit what they call a photosynthesis irradiance curve. And this has 2 essential parameters. The initial slope, which is often linked to the efficiency of the phytoplankton in primary production, and the assimilation number, or known as PB max, which is often referred to as the capacity of phytoplankton for primary productivity. And we also have this parameter here, IK, which is essentially a ratio of these 2 parameters and gives us an index of the light levels at which the phytoplankton are acclimated at. Now, this is the mathematical formulation of that curve I showed you in the previous slide, this green curve here, where our normalized production PB is modeled using this exponential equation, PB max here, that assimilation number, and alpha B here, and then our irradiance up here on the equation. So assimilation number of the light saturation curve, PB max, the initial slope, and the level of light. So going back again, light I, PB max, maximum, and then this initial slope alpha B. Okay, so one thing to bear in mind is that many of these parameters and variables vary with time and with depth. To keep things simple in this example, our PB max is varying with depth here, and so is our alpha B, and our light irradiance is varying with depth and time here. Now, of course, we could look at spectral variations in light. So if we had a spectral primary production model, these 2 parameters would also be wavelength dependent, but we're keeping things simple here. So we're dealing with light in the form of PAR, photosynthetically available radiation, so light integrated over the visible part of the spectrum. Okay. So if we want to know the primary production in units of meters squared per day, we have our same equation here, the photosynthesis irradiance curve, PB max and alpha B and light. And now, of course, we're converting back to our primary production here. In order to do that, we need to scale to our biomass, right? Because this is the normalized primary production. Our biomass also is going to vary with depth. And then we integrate over the day length D and here I'm using the euphotic depth, but you could integrate further down into the water column, but just bear in mind that as you get below the euphotic depth, there's not much light for photosynthesis anyway. So just for the purpose of this simple example, we're going to integrate down to the euphotic depth. Do bear in mind that this simplification does not account directly for spectral variations in light-driven photosynthesis, as I just mentioned before, and assumes the photosynthetic parameters and the biomass do not change over the course of the day, when, of course, in reality they can. Okay, so how do we do this then from satellite? Well, first of all, we need to know the day length, right, as part in the first integral here. So we can get a good proxy of day length simply as a function of latitude and time of a year. So we can model that pretty well. The next thing we need to know is the depth of the euphotic zone, the 1% light level. And again, this can be estimated pretty well from satellite ocean color data, actually. For example, using estimates of the diffuse attenuation coefficient, and the euphotic depth is now a sort of standard product actually produced by many space agencies. So day length and euphotic depth we can get quite well from satellite. Okay, so our next parameter we're looking at is PB max. Now this is a bit of a tricky one. It's not so easy to derive directly from satellite ocean color data, and as a result, there are many different ways at which we've tried to produce maps of PB max, or the assimilation number, using different techniques, which I'm going to go through in a minute. The first technique I'm going to show here is using other environmental data. So there's work that has looked at the relationship between PB max and sea surface temperature and irradiance. This is built on sort of some of the early work initially by Epley and others. We can get very accurate estimates of sea surface temperature and PAR from space. And so if we know that from satellite, we can use these models to sort of predict what PB max is from satellite and then feed them into our primary production model. Another approach, actually this was a nice paper published by Trevor back and colleagues in 2008, is to use large in situ data sets of PB max and then sort of develop techniques that look into this data set and find, for a given pixel, similar conditions. So if we can get chlorophyll and temperature in a pixel accurately from satellite, we can then scrutinize that in situ data set to find conditions with similar chlorophyll and similar temperatures, and then find or search for an adequate PB max, a measurement that was conducted under those conditions, and assign that to a satellite pixel. Now, another technique is thinking about phytoplankton community structure. Now, next week, I believe Heather Bowman will be talking about that in Module 11, about phytoplankton community structure. But what we typically see is that different communities of phytoplankton in the ocean photosynthesize it in different rates. And so the parameters, both the initial slope and PB max, vary depending on the size, in this case, in these examples shown here, but also the taxonomic composition of phytoplankton in the ocean. So if we know that, if we can predict what the type of phytoplankton is in the water from space, we can really improve our assimilation number estimates. Now, one of the another approach that has had a lot of success and sort of goes back to that map I showed you by Alan Longhurst, the first global satellite estimates, is to use biogeographic provinces or biogeochemical provinces. And so these are regions of the ocean which are relatively homogeneous. And so if we have data collected in that specific region, we can use these, we can average that within the region and feed that into our satellite primary production models. Not only regionally, but also seasonally as well. So we can look within a given province at how the PB max varies over the course of a season and feed that into our satellite primary production estimates. Similar to PV max, alpha B is another parameter of this satellite photosynthesis primary production equation that's a little bit tricky to get at. Though there are some nice recent work showing how well actually estimates of light can help. But these again, just three ways at which we can estimate alpha B. So this initial slope, the efficiency of the phytoplankton, we can estimate that parameter and feed it into our primary production estimates. Again, using the nearest neighbor technique I mentioned before, or looking at phytoplankton community structure, or thinking about aggregating the in situ data sets into different biogeochemical provinces. Now, there is a lot of exciting work actually going on, and I know there's going to be a module 16, I think, looking at artificial intelligence, but a lot of promising work showing how well AI-based or machine-based learning may be able to help in our estimation of these two photosynthesis parameters, alpha B and PB max. Okay, so let's say we solve that. We now need to know the light environment. Now, fortunately, we can get very good, accurate estimates of photosynthetically available radiation from satellite data. And so we can quantify the light environment just above the sea surface. Now, if we know that and we can make good approximations of the diffuse attenuation coefficient, another key variable that we can get from ocean color satellite data, we could begin to propagate that surface light down through the different depths in order to model our light environment accurately, driven by satellite observations. And of course, this diffuse attenuation coefficient may vary with depth as well, right? So in some regions of the oceans, we have a deep chlorophyll maximum, like I'll show you in a minute. So we even have models that can consider depth variations in diffuse attenuation coefficient in order to improve our estimation of the light environment in these models. And finally, our biomass. I talked about a bit earlier about this importance of scaling to phytoplankton biomass. So we can get chlorophyll concentrations very nicely at the surface from space. But of course, that's only at the surface. If we want to know what's happening further down the water column where the satellite can't see, but whether there's still light available for photosynthesis, we need to be able to model or to extrapolate those surface observations down to depth. And there are various empirical, semi-empirical, and machine learning based methods that are now being introduced, driven by not only satellite observations, but also in the increasing use of ocean robotic platforms like biogeochemical Argo floats in order to help that extrapolation of the surface fields down through depth. Okay, so if we have all that information, then on a per pixel basis, we can begin to quantify a global primary production. This is just an example of an estimate of primary production from May 2004. So what we can see here is we're going into the spring bloom period in the North Atlantic, and we see very much higher rates of primary production in the North Atlantic, where the day length has got quite long and where we have the phytoplankton spring bloom. Of course, we can use satellite observations like we showed earlier in Tom's nice video of chlorophyll concentrations. We can do similar things for primary production. So we can begin to look at the seasonal variations in primary production. This is an example from a model where we estimate the primary production from large phytoplankton. And we can sort of see quite clearly in this image in the Atlantic Ocean how the seasonal variations of phytoplankton primary production varies over the seasonal scale. Okay, so in terms of validation, one of the critical things with all satellite products is we need to be able to have good in situ data sets on primary production to evaluate and validate our satellite observations. There was a lot of effort by NASA in the sort of mid-90s through to sort of, I think, 2011, where they ran a series of primary production round robins. They tested different primary production models and validated them. And it's commonly assumed nowadays that the uncertainty in primary production is around 30% in log10 space, around 50% in linear space. Though I have to say there's a lot of work going on now improving our estimation of uncertainty in primary production. And I know Gemma, who's on the panel, has been doing a lot of nice work on that recently. So this is very much a moving field. And this is just an example of sort of our most recent estimates of global primary production from a very nice paper by Gemma in 2021 using the Ocean Color Climate Change Initiative data. And they quantified global primary production to be in the region of around 48 to 53 gigatons of carbon per year. Again, very similar to that estimate of Longhurst back in 1995. Okay, and so one other thing to mention, we talk mainly about primary production, but there's also increasing work looking at how we estimate that export production, the amount of production that's in the surface layer that gets exported below the euphotic zone. And there are techniques that people have developed to do this. This EF here is what they call the export production to primary production ratio. And the people have developed empirical approaches to estimate this, driven by satellite temperature and primary production estimates. So you can convert your satellite primary production estimate into an estimate of export production. And a lot of work going on this topic in the European Space Agency BICEP project. Recently, they've been testing an empirical approach by Dunn et al. In 2005. And that brings me nicely on to the last point of the lecture, which is just to illustrate some of the resources on primary production that are available to the community. These are 3 data sets that have recently been produced in the BICEP project. There's a monthly global primary production data set based on the work by Gemma in her 2021 paper. Monthly primary production from 1998 to 2020 at 9-kilometer resolution, all freely available. Additionally, we've just also in the project published a monthly global export production product as well. Again, going from 1998 but this time to 2019. And there's also products on phytoplankton biomass, not only in terms of chlorophyll concentration, which we can get from the Ocean Color Climate Change Initiative, but also within BICEP, the phytoplankton carbon concentration. So estimates of phytoplankton carbon. Also, some other resources here. NASA run a site on ocean productivity, which has some other primary production products. Again, really encouraging you to go and look at this in situ aquatic primary production field protocol series. It's really excellent. And there's a link directly down there on the resources button. And also just to let you know that we've recently had an IOCCG working group approved in March 2023 on primary production. And so we hope to be producing one of these nice IOCCG reports in the coming years on primary production. So that's it for the lecture. Just a very brief overview. We've talked about our definition of phytoplankton biomass, how we measure that in the sea, dealing mainly with chlorophyll concentration in this lecture, how we measure that fluorometrically using high-performance liquid chromatography or in vivo spectrophotometry, how we measure phytoplankton biomass from space, linking back to some of those early modules that you would have learned on relating the color of the ocean to the chlorophyll concentration. Definition of primary production, this conversion from inorganic carbon to organic carbon through the process of photosynthesis, why it's important for the carbon cycle for fisheries, how we measure that in the sea, going back to some of the very early techniques of the light and dark bottle method, all the way through to the various methods they use today. Again, didn't go into that in too much detail, but the resources are here available in these slides. And then a bit on how we measure it from space using a simple available light model and some resources. So at that point, I stop. I think I've been just over the hour, so I think it's a nice point to stop there.
Thank you, Bob, for an excellent lecture. You're right on time, I think. It's very well done. There are loads of questions, so I think we'll keep you busy for a little while. and I'm sure the panel members can help as well.
I hope so. I can't think of a better panel to be with me to help with these questions, to be honest. So feel free to jump in.
Yeah, lots of compliments also in the chat. Actually, I'm going to start with an easy question, maybe, which is quite a fun question. So one of the first questions that came up is, do you need to be a biologist to investigate what you presented?
I am not a biologist. I would not consider myself as a biologist, so I don't think you do. I mean, I think what's really great about primary production, satellite primary production, is it's a very interdisciplinary part of oceanography, right? Because you have the physics, you have the light, you have the retrieval process of ocean color. A lot of physics going in there. Physics, the physical, chemical, biological interactions in the ocean dictate the primary production, right? And so I don't think you have to be a biologist at all. And what's fun sometimes is working in an interdisciplinary team with chemists, with biologists, and with physicists in order to improve our understanding.
Thank you. I could agree actually to that. So I'm just gonna go through the questions following the topics of your talk. So you started with measuring biomass at sea. So there were a couple of questions related to that. There was one question about the fluorometer and whether you could explain in a bit more detail how it works and how you would measure chlorophyll A.
Sure. So I'm not an expert on fluorescence, actually. So maybe that somebody wants to jump in and chip in as well. But in principle, fluorescence is a process. It's sort of an inelastic scattering process, right, where the phytoplankton absorb light at one wavelength and then re-emit it at another. So how the fluorometer works is you send a beam of light at one wavelength, typically I think around in the blue, where the chlorophyll absorption is highest. It then gets absorbed by the phytoplankton and then re-emitted at a different wavelength, typically around I think 680 or 683 nanometers. Now, there's two ways I mentioned, I think, that they do this: the in vivo and in vitro method. In vivo, they send that light source directly at, the fluorometer's on the CTD and it's sending that light and measuring the fluorescent signal as water flows past it. And there's also the extraction method where you filter the chlorophyll. You then essentially put it in a chemical which extracts all the chlorophyll into the solvent. And then you measure that the fluorescence of that solution. But I didn't go. I avoided going into more of the biochemical process behind photosynthesis itself. So if anybody wants to mention that, they're welcome.
Thank you, Bob. There's another question about measuring biomass at sea. And that is, how do we avoid the chlorophyll A concentration changing through photosynthesis between sampling and the measurement?
Oh, yeah, that's a good question. Yeah, so it's not so much of an issue, I guess, when you're doing the in vivo measurements, right? Because you're measuring the instantaneously in a way. And even with the in vivo spectrophotometry, similar, that the water's flowing through the ACS and it's measuring the absorption instantaneously in a way. But yeah, when it comes to measuring using filtration-based methods, HPLC or using a benchtop fluorometer, then yes, there is you need to be careful, right? So your Niskin bottle snaps shut at depth. It gets brought to the surface. It's immediately in a dark environment, whereas it might have been in a light environment. So immediately things are going to start happening. There's then going to be various points at which, as you move along that measurement process, where things can happen, right? Your sample can start settling in the Niskin bottle, so you can get it can be not quite fully mixed, right? So depending on when you sample from that Niskin, you might be getting a different part of the measurement. You ideally want to do things, I guess, as quickly as possible. There are protocols that the community have put together on this. So I would recommend, if you are making those measurements, to follow those protocols quite carefully. But yes, by all means, things can happen from that point at which you shut the Niskin through to when you make. I think Osvaldo.
Yeah, Osvaldo can add. I can also add that most protocols dictate that you work in the dark as much as possible to prevent things from happening once you've sampled. Osvaldo, do you want to add?
Yeah, well, after many cruises experience at sea, in fact, the pigment, the chlorophyll A, once you extract it, I mean, it's relatively stable. As two things you have to worry about. The one you mentioned, Gemma, light, so not expose the pigments to high light, so you work in dim light. And the other one is temperature. So you have to freeze your sample as soon as possible or put them in ice. So those are the two things when you are at sea or in a coastal system or whatever or lake, whatever you're sampling, is to extract your pigments in darkness and put them as soon as possible into ice or liquid nitrogen or whatever. So.
Thank you, Osvaldo. There's another question, whether there are observations for other components of organic carbon, such as DOC or POC or zooplankton. I think you already touched upon this a little bit. There's definitely in situ data available, but also satellite products, at least for DOC and POC. And I think if people want to know more about that, I would suggest they have a look at the BICEP project or some of the NASA products that are also available. Then there is another question, which I'm not sure if you've talked about, but there was a question about chemtags that's often used in combination with HPLC analysis to determine phytoplankton taxonomy. So what are your thoughts about that? Actually, this student asked, some professors like it, some professors don't like it. So I think they're just looking for your opinion. And I think maybe Heather will also comment on it next week.
Yeah, I mean, I think one of the techniques I mentioned, HPLC, allows us to look at the whole suite of phytoplankton pigments. And that can give us useful information, not only on the biomass, but also on the community composition of the phytoplankton, because certain phytoplankton taxonomic groups have higher concentrations of certain pigments than others. So I'm thinking off the top of my head of diatoms have a pigment called fucoxanthin. They have a lot of that pigment, but that pigment, unfortunately, is also in other phytoplankton as well. So you get to this point where many phytoplankton groups can share certain pigments. So it can become tricky to differentiate between different taxonomic groups based purely on their pigment structure. One of the nice things about HPLC is it's a technique that's been used a lot over the past sort of 20 or 30 years. So there's these huge global data sets of HPLC pigments, which from a modeling perspective, from a sort of satellite perspective as well, it's become quite a powerful technique for developing phytoplankton community structure-based satellite algorithms, which I think Gemma mentioned Heather's going to talk about a bit next week. And there's various ways you can look at these pigments. Chemtags is one method. Another method is what they call diagnostic pigment analysis, which is just directly relating certain pigments to different taxa, different size classes. I mean, I would say that if you want to look at community composition, having as many techniques as possible to do that, the better, right? So if you're making HPLC measurements, but you also have flow cytometry, you also have microscopy, you also have I don't know, particle size distribution data, then that can really bring more confidence to the community of phytoplankton you're looking at.
Thank you, Bob. TJ, do you want to add?
Oh, I was just going to add that I think fundamentally the validity or usefulness of chemtags comes down to, one, whether or not the suite of pigments that were sampled is consistent across all your measurements, because when you compare HPLC data from different cruises, sometimes they have measured different sets of pigments because the set of standards you need is quite expensive and all these sorts of things. And the other thing that goes into the uncertainty in those outputs is basically the stability of the pigment ratios that you allow for those different groups. So as Bob said, it's a very useful tool, but you just have to understand where the issues can lie with it and then look at the data set in detail.
Thank you, TJ. Related to this and moving on to measuring biomass from space is a question on whether we could measure phytoplankton from space and functional types.
So yeah, that's a good question. Actually, there is a very nice IOCCG report led by Schuber, actually, I have to say, published a few years back now, specifically on this topic, that I would recommend going and having a look at. So how we detect phytoplankton functional types from satellite data. There are techniques that have been developed to target phytoplankton in the context of the chlorophyll concentration, but also actually cell number and carbon. Within the BICEP project, there are phytoplankton carbon products that are produced that are available for phytoplankton, as well as nano and micro phytoplankton. It's a rapidly evolving field, actually, and we're hoping that with the advent of hyperspectral ocean color data, we may be able to improve our estimates of phytoplankton community composition. Typically, two approaches to it. One of them is to try to directly detect the different taxonomic groups by looking at the optical signatures in the satellite, which is tricky to do, but it's a direct approach. But there's also a variety of indirect approaches that relate the community of phytoplankton in the water to variables that we can derive confidently from satellite, like the chlorophyll concentration or like some environmental parameters like temperature or light.
Thank you, Bob. Another question I think was related to when you were talking about open ocean algorithms. So a question was whether case 1 and case 2 waters, that classification is still used, and if not, what else should we be using?
Yeah, so this was a classification, I think, developed by Morel and Pur in sort of 1970s. It's a very broad classification. And I think there's a lot of exciting developments in these types of classifications, and Tom's probably a very good person to touch on with that in the context of optical water types. So I know within the CCI project that Schuber and Tom and others have been working on having satellite ocean color algorithms that are designed for specific optical water types. Now, the number, I think in the CCI project they have 16 different types of optical water types rather than just case 1 and case 2. So they have a whole transition of optical conditions in that. I still like the classification because I'm a kind of simple guy, I suppose, and I like to keep things quite simple. And I also like to study a little bit of the history and there's a lot of use out of that classification, I would say, over the past sort of 30 years or so, 30, 40 years. But yes, nowadays there are all these suite of methods that are being used to derive optical water types, which can then help us improve our algorithms.
Thank you for that. So there are some questions about zooplankton and higher trophic levels, which I'm not sure if you can answer. But the questions are related to whether we can observe organisms that feed on primary producers from space.
I could have a
go. And whether that's related to phytoplankton.
Yeah, so yeah, there have been approaches proposed to estimate or observe directly higher trophic levels like zooplankton in the ocean. One of the techniques I recall was by a guy, one that comes to mind, Patrick Stromberg, I think, had an approach when I was working at PML that basically looks at the essentially the particle size distribution in the ocean. So if we know what the particle size distribution is in the phytoplankton, we can then extrapolate that into the higher trophic levels. So yeah, essentially, and this goes back to work that Trevor again was doing, you know, in the 70s, 60s and 70s and 80s with looking at ecosystem structure in the context of the normalized biomass spectra work. So there have been approaches that follow kind of that line. More recently, there have been techniques that have tried to at least directly observe some zooplankton from ocean color. David McKee was involved in some work actually that showed that when you get very high accumulations of zooplankton in the surface ocean, it actually has an optical signature that you can retrieve from satellite. There's also been work looking at LiDAR actually. So looking at the because with LiDAR, it's an active system. It can operate during the day and at night. And of course, if you know the cycles of zooplankton in the ocean, open ocean, they have these diurnal cycles where they come up during the night to feed on the phytoplankton and then go down at depth in order to avoid predation, I think, actually. And so by looking at the backscattering differences from LiDAR between day and night, they believe they've picked out signatures from zooplankton.
Thank you, Bob. I think you did a good job there. So continue on to measuring primary production at sea. You talked about GPP and NPP, and there's quite a tough question here. How can we ever quantify NPP? How do we distinguish between autotrophic and heterotrophic respiration? Do you want me to help here?
Yeah, I might just take a little breather and come in if you want to have a crack at each other first.
I think this is one of the big challenges. So there are lots of techniques how to measure primary production at sea in the field. And within the community, there's also always a question, what do we measure, NPP, GPP? I think if you want to distinguish between autotrophic and heterotrophic respiration, you have to make assumptions. If, for example, you do a 14C incubation for 24 hours, you could assume that the dark respiration that you measure is only associated with phytoplankton, or the bulk of it is phytoplankton. And then you could do additional measurements, for example, oxygen respiration measurements for your total community. And you could then distinguish the heterotrophic respiration. So it might require a combination of measurements to determine those different components. But it's always a big question about what you are exactly measuring. And there is the IOCCG protocol that came out last year on measuring primary production in the field, does give a good introduction and overview of that. So that might be worth reading for people who are interested in that. So hopefully that answered that question. So there are many questions. There was a lot of interest about your topic of primary production from space. So the first question that came up is if we ever have negative primary production, and if so, when?
Negative primary production. I mean, of course, you have respiration in the ocean, right? Which is the inverse process, essentially. And there's what they call the compensation depth when you move down the water column, where your respiration becomes higher than your primary production. But negative primary production. Don't know if you want to comment on that, Shuba.
I think Bob, this might have referred to your plot where the curve of the PI went below zero.
Yeah,
and that's the point at which your respiration is larger than your photosynthesis.
Absolutely.
So that's what happens at night when your radiance drops below that critical level. You effectively aren't photosynthesizing as much as you're respiring, so your production becomes negative, I guess, at that point. I think that's what that was referring to.
But not your primary production, right? Can you hear me?
Yes, we can hear you, Shubha.
Yeah, so primary production by definition is a positive process, so it can't go negative. But respiration in combination with primary production, you can get a negative value. And most well-run production experiments will include a dark bottle for measuring respiration. So when you see a negative value, that implies that you are looking at what's happening in the dark, and that is dominated by respiration, which is the inverse process, as Bob mentioned, to primary production. But that is not primary production. It's a difference between production and respiration.
Thank you, Shubha, and also TJ for clarifying the question. I did not realize it was related to the figure of the PI curves. So we continue on with some more questions on this topic. So one of the questions is, if primary production is related to phytoplankton biomass, does it make sense that phytoplankton blooms have high primary production? And then a related question to that is, when there are high levels of primary production, is this harmful for ecosystems?
Okay, so I'll start off, I guess, but by all means, please help me along along the line. So, I mean, I think it was useful in the lecture to separate out biomass and primary production, right? So you could have a situation where your biomass normalized primary production is low, but you have so much biomass that your primary production is actually still quite high. So often you could have a bloom where your normalized primary production is high and your biomass is high, and then your production is really high. Or you could have a bloom where your normalized production is low, but your biomass is still high. And so relative to the background levels before the spring bloom, your primary production is still quite high. In general, when you have a phytoplankton bloom, you have increased production, I would say. But then there's also a strong gradient, potentially a stronger gradient in a shift in the vertical structure of the primary production as well, because a lot of your light is getting absorbed at the surface, and so you might have less primary production at depth. But if you integrate that, normally your primary production is higher during a spring bloom. I'm not sure if I've just muddled that a little bit, but I hope you get the point. And yes, by all means, if you have a very high, big phytoplankton bloom, you have a lot of primary production, you think that organic matter is going to be sinking out of the water column, you're then going to have potentially a lot of consumption of oxygen at depth, remineralizing that organic matter, and that can result in oxygen minimum zones as well. The phytoplankton bloom can modify the thermal structure of the ocean, actually promoting stratification. And yes, depending on the species of phytoplankton, you can get production of toxins and harmful algal blooms.
Thank you, Bob, for that answer. There are a couple of questions related to the equations and the models that you showed. So one question was whether the concept of estimating primary production from satellite could also be applied to macro algae.
Yes, I believe so, right? Essentially, the fundamental process of photosynthesis is similar between plankton, between macroalgae, between terrestrial plants, right? So yeah, I mean, if you can quantify the biomass of macroalgae using satellite observations, and you know their photosynthetic rates, then by all means you can estimate the primary production of macroalgae. I say that I've never done it, and I'm a little bit ignorant of of detecting and estimating primary production at macroalgae. I recall a nice talk by Xiumin Hu, I think, at the Ocean Carbon from Space workshop, where they looked at quantifying primary production from sargassum, I think, actually, in the North Atlantic, and looking at their relative contribution of sargassum to primary production by phytoplankton. So yeah, that was there is work going on in that field.
Thank you, Bob. Another question is whether absorption by phytoplankton would be a better model input than biomass for the estimation of production.
Yeah, that's a good question, actually. I think I alluded to again a nice paper. I thought I always really liked it, Shuba, your paper with Trevor in 2007, which basically laid out the different ways at which we can model primary production, kind of with reference to satellite. I used an available light model, which is biomass driven, but there's also absorption-based models as well. So rather than having biomass with chlorophyll, you have absorption by phytoplankton. Now that can be quite a good model to use, particularly in optically complex waters, where our estimate of biomass of chlorophyll can be in error. Whereas you separate out the optical properties by phytoplankton, by CDOM, by other material. And so you can use the phytoplankton absorption component in that. I think one of the tricky things with the absorption-based models is quantifying the amount of absorbed light that's then used in photosynthesis versus other processes that happen within the phytoplankton, like photoprotection. But yeah, there's a lot of people have been using absorption-based models rather than biomass-based models. And I think there's pros and cons to all of these methods. And as Shuba showed nicely in her paper, they're fundamentally very similar, actually. You can convert between them.
Thank you for that answer. You talked about PI parameters and PI curves. So there's a question on whether there's a systematic error in those measurements. And I would like to explain a little bit more. It's quite a long question. So PI parameters usually measure something between MPP and GPP, depending on where you are in the light curve. And the same goes for DOC. So more DOC would be excreted at higher light levels. Is there a systematic bias or error in those measurements? But yeah, maybe it's a better question for Heather, but I'd like to ask you anyway.
Yeah, so I'll allow others to jump in if they feel up for it with this one. I mean, it's hard for me to comment, to be honest, because I haven't made many measurements of P&I curve parameters before. My understanding actually is that they are the more desirable method to use to get at primary production in situ, and also because the parameters have a direct relation to how we model primary production from space. But they are short incubations, right, of the order of 1 or 2 hours normally, unlike the longer on deck or in situ incubations, which typically are in the order of 12 to 24 hours. Those incubations, those longer incubations, are thought to be closer to net primary production because you account for the respiration component better. And it's hard to know, I guess, within a 2 hour period the information on the amount of respiration that's occurred in the sample, but also the DOC excretion as well. Not only it might happen in terms of light, but again, I think that's a pretty hard thing to quantify with a 2-hour incubation.
Thank you, Bob. Shiva, do you want to add anything?
Yeah, just to add to that. Remember that in all these labeled carbon experiments, for respiration to have a signal, there is a finite time required. The labeled carbon has to be first taken up by phytoplankton, and then they have to respire such that the carbon leaves the phytoplankton. So if you give it enough time for phytoplankton to take up the labeled carbon and then to respire it back into the system, then you are moving towards net primary production measurements, because you are automatically correcting or incorporating the effect of respiration. But that is the advantage of PI experiments. You try and keep the experiment as short as possible so that you don't give the phytoplankton enough time to respire the labeled carbon that they have acquired. So as Bob said, that is the general principle, that the shorter you keep your incubation period, the closer you get to gross primary production. The longer you keep it, more you are into the domain of net primary production. But if anybody who has actually worked in this area will tell you that it is almost never possible to meet these ideals if you want to use a labeled carbon method. And if you don't want to use labeled carbon and you use other methods, then very often the incubation times are longer, and then you approach new production as opposed to even net primary production. Maybe Osvaldo can also add something to this discussion.
Osvaldo, oh, you are muted, I think.
Oh, am I? No?
Yeah, no,
Okay.
we can hear you.
Well, I don't have much more to add, Shubha, and the audience. I think you have pointed out the things that we need to consider. I don't know if anything in particularly that you were thinking of that I could add, but I see that things have been covered and answered in particular. I don't hear you, Shubha.
I muted myself. I thought none of us touched upon the fact that from a practical point of view, the difficulty lies in the size structure of microbial communities. There is an overlap between phytoplankton and bacteria, and between phytoplankton and zooplankton. And that makes it very difficult to say, or to effectively isolate phytoplankton and only look at what they are doing. So practically, we have all these difficulties.
Yeah, that's you're absolutely right that there's overlap between size classes, but the nice thing about people that work with phytoplankton is that first we have pigments, that gives us an advantage and the ability to separate what is in principle autotrophic versus heterotrophic. So that is definitely a powerful thing when we work with primary production, is that our methods are based on having a good estimate of biomass through pigments, and that obviously so that's the critical part, and I think that's where satellite oceanography has been so powerful, is because we are able to measure one of the variables that contribute most to the variability of primary reproduction, which is phytoplankton biomass. And I think that's it. Always we need to stress that you know the successful of all these approaches is based on our ability to measure phytoplankton biomass, you know, quite accurately and at the global scale. If we didn't have that, we would be in much more trouble. So then, of course, when we know that there are other microorganisms, other processes going at the same time, and that they overlap, you know, with other organisms. But at least in the ocean, where you have oxygenic photosynthesis, you can separate processes that are, you know, fixing carbon, and we can have good estimates with that. So I don't think we need to, I said, to put too much emphasis on the difference, but actually we should really put effort in try to look at the general rules, the things that we can predict better, and then try to see what are the parts that we cannot account for. And I think that again the field is moving in the right direction in saying, okay, well, you know, we have general estimates, or we had global estimate where at the beginning we only had was phytoplankton biomass. But now, you know, with the ability of being able to differentiate different different size classes, we are starting to get much better estimates of primary reproduction. So in that sense, I think that this overlap thing is a second order problem, and that you know, that we're fortunate that we are being able to deal with the first order problem, which is to you know, to estimate the variables that Bob was pointing in his lecture. You know, I think that's the strength of the remote sensing of primary production.
Thank you, Osvaldo, and also Shuba, for your answers. There's one more question about the PI parameters, and I think I will be the dedicated person to answer this. So the question is whether averaging the PI parameters over regions and seasons would mask longer term trends. And this in sort of relation to climate change. If anything would change in response to climate, would that not be the photosynthetic parameters? So I can comment a little bit on this. So in the primary production model that we use at PML, we do assign PI parameters per province and per season, and this is relatively coarse. But it's the best that we can do at the moment. So we make use of a large in situ database that Bob has showed, which now has, I think, over 10,000, maybe 12,000 measurements in it, which sounds like a lot of data, and it is a lot of data, but we still face patches of ocean where we don't have any data. So we realize that this assignment is quite coarse. It does not change between the years, for example. So in response to climate, we know that the PI parameters are not changing over time. So this is one of the challenges we are currently working on, and Bob has already mentioned this as well, that it's important that we work on the assignment of the PI parameters, especially in climate research. So I think it's a very relevant and smart question to be asking. So I hope that answered the question. There are a couple of other questions. So one of the questions is, what is the benefit of looking at primary production? Why wouldn't we use chlorophyll A instead?
Shall I come in here?
Yes, please.
Okay, so I hoped at least in the lecture I kind of touched on this question a bit by separating out the biomass and the primary production. So I think if you recall on one of the earlier slides, I showed how minuscule the biomass of phytoplankton is with respect to the terrestrial plants, right? On the order of 0.2 to 1 gigaton of carbon. Gigaton of carbon, I think. Compared to 450, 500 gigatons on land. And yet, despite that tiny biomass that we can quantify from our chlorophyll or from our carbon measurements, you're getting equivalent amount of primary production by phytoplankton as you do on the land. Now, as we explained in the equations, actually, some of the very early techniques that tried to estimate primary production from space did involve just converting the chlorophyll concentration into an estimate of primary production. But that ignores any, one of the most important things in primary production, which is light, right? So these the extension of that was to then incorporate how much light, the importance of light in that estimation of primary production. So, for the same chlorophyll concentration, basically the primary production can be very different depending on the light environment, right? If you've got a long day with a lot of light, your primary production is going to be much, much higher than it is if you're, in the polar regions and you've got an hour of light midwinter or something, if you're lucky. So that, I hope that at least gets at the point, and it's primary production that drives the ecosystem, right? It's that production, that interest on your capital or whatever, that then feeds the ecosystem.
Thank you, Bob, for that answer. Shiva, are you raising your hand or no? Okay, good. So we're getting close to 2 o'clock in the UK, so I'm gonna sort of round up with a question. Well, there are a couple of questions related to what happens if phytoplankton is under stress. Does the physiology change? So there's mention of stress conditions such as temperature, warming, but also changes in CO2, ocean acidification. There's photoinhibition. So I've termed that as stress conditions. Does that affect physiology? And then does that affect primary production and the estimate of primary production from space?
Absolutely. I think, yeah, that's a nice example of the photoinhibition. So we didn't really talk about that in this lecture, but you can get the presence of photoinhibition in the ocean. So this is where, if you recall, we looked at the photosynthesis irradiance curve, and it reached a maxima, or the assimilation number. Now you can get conditions where the light continues to increase, the production starts to drop off, and can be quite radical, actually, depending on the species of phytoplankton. And so that is a prime example where phytoplankton are under stress, light stress, and therefore are not producing, they're not being as efficient primary producers.
Thank you, Bob. I just want to ask one more question, which was related to the uncertainties. So there's a question on whether there are products available for high latitudes, such as Svalbard, Spitsbergen, or Greenland, where conditions are very different. And how do we deal with the uncertainties in the environment, so as glacier melt, icebergs, and things like that?
Yeah, it's a shame Heather's not around, actually, isn't it? Because she does a lot of work on primary production in the Arctic. I don't know if anybody else wants to come in. Maybe Shuba want to come in and help with that. I think just very briefly, yes, there are many scientists working on estimating primary production from space in the Arctic, and you have a number of complexities and intricacies. Tom, again, is another person who's done a lot of work on primary production in the Arctic. You have different vertical structure in phytoplankton biomass, right, Tom?
Yeah, I mean, I think what it comes down to is working out which is your biggest error in that case. So yes, there might be an issue that they have quite a unique photophysiology for the light environments that they're adapted to. But I mean, in Arctic environments, if you don't account for under ice blooms, I mean, you can get diatoms stuck to the bottom of sea ice that can be extremely productive, but they're in a very thin layer, and they're quite important for the kind of ecosystem, especially as the light levels start to increase, but before the sea ice melts back. Then you've got different populations that are forming your ice edge blooms. So as the ice retreats, you get the stratification, and you get these strong blooms on the edge of the sea ice. And then, yeah, you've also got issues of, is the chlorophyll estimate wrong anyway, because you've got glacial outflow and glacial flower in the water. So in each of those different environments, there's a different major source of error. So in the sea ice, it's whether or not you're accounting for that under ice layer. In the ice edge, it might be whether you've got the photophysiological parameters right. And in the glacial outflow area, it's probably that the chlorophyll concentration is significantly affected by the sediments in the water, maybe, or the optical properties. So I think it's just working out which of your errors is dominant and trying to minimize that in each of those environments is probably the biggest source of concern or thing you should work on.
Thank you, TJ. That was our final question. I would like to wrap up. Thank you, Bob, for your lecture and answering questions, and also to the other panel members. We've had some really challenging questions, I think. So it seems that our students are learning, also learning how to ask questions. So this is very good. Thank you also to everyone for being here, and we're looking forward to seeing you next week when Heather Baumann will be presenting.